All right, then let's go ahead and get started with the second presentation. This one by Alec Lammens on intuitive explainable artificial intelligence for molecular design. So please.
Thank you so much. So as Brandon said, I'll be presenting some of the work I've been doing on providing intuitive explainable artificial intelligence for molecular design. So I'm Alec. I work for the same group as Andrea. And So that makes a brief theoretical outline of what I'll be presenting today. So I'll start with some background information on explainable artificial intelligence, including some of the nomenclature we use and some of the different kind of methodological categories. Then I'll show you some of the most well-known XAI methods and how we've adapted them to molecular modeling specifically. And I'll finish off with some concluding remarks and a future perspective, as Andrea has also already highlighted. So first of all, a bit of background. So machine learning models can generally be characterized as white box or black box models. Well, this is a bit of a generalization, and there's a whole spectrum, including gray box models. For the presentation, I'll stick to this simple binary comparison. White box models are said to be inherently interpretable. What do I mean by this? We could simply look at the inner parameters of the model to see why a specific decision was made. So, for example, the humble decision tree. We could go down the decision tree, go to each specific split, and see why a specific prediction was made. However, of course, these models are quite simplistic, and if you have a lot of data or intricate data patterns, the simplicity will ultimately lead to a low accuracy. So especially nowadays, these kind of models are not generally used. However, of course, if your setup allows for it, it's always good to check whether a very simple model works, because it's inherently explainable, which is a big advantage. On the other end of the spectrum, we have black box models. So these are the type of models that focus on getting as much predictive power as possible, but usually at the cost of becoming increasingly complex. So these are, of course, all your deep learning frameworks, ranging from deep neural networks to graph neural networks. Generally, we cannot simply look at the inner parameters of a model to see why a decision was made. These models are far too complex. And this lack of interpretability is usually what hampers a practical application. Even though they're very powerful and flexible, it's hard to have an end user trust a prediction if you cannot explain why such a prediction was made, especially in our field of chemistry, where we have to convince chemists to generate the molecule, to synthesize the molecules we propose. So this is where explainable artificial intelligence comes in. So simply put, we try to provide explainability from black box model predictions. Or what I do mean with explainability, simply explain why a specific decision was made. And for this, there's a large focus on post hoc XAI methods. So this means we already have a fully trained black box models, and then you make the predictions, and you explain these predictions with a post hoc explainer applied to this already fully trained model. One of these the big problems is that explanations might perfectly capture the learning characteristics or the internal parameters of a black box model. However, this does not mean that the resulting explanation is interpretable to us as the end user. This is where we have to go from explainability towards interpretability, where we render these resulting explanations interpretable to us. A bit of a note is that the definitions for explainability and interpretability are often sort of interchangeably used, and depending on which line of papers you aim, these definitions might even switch meaning. But for the sake of this presentation, I'll stick to these definitions. But how do we achieve this interpretability? I think for this, we should render explanations clear and easy to understand. I think one of the best ways to approach this is by tailoring existing XAI methods to provide explanations in the application domain. And this is what I want to be focusing on today, especially for molecular design. So to provide a basis for the comparison of different XAI methods, we have a set of characteristics that we would like them to have, which are called desiderata. So it's basically to provide a communal nomenclature we can use when we talk about different XAI methods. So examples of these desiderata include robustness, basically meaning that explanation should be robust to minor data perturbation, or stability, which is related that if you have very closely related data points, you would like the explanations to be similar, which makes sense. Other examples include completeness, that explanation should account for all of the important features that have had a significant impact on the model outcome. And the one I want to focus on the most today being domain applicability, that the explanation should be interpretable in the application domain. So we have a lot of different XAI methods that have been proposed over the years, and you could classify them in any number of ways, but I'll focus on 3 types of XAI method for today, being feature attribution methods, rule-based methods, and example-based methods. Feature attribution methods aim to quantify the contribution of every individual feature to the final prediction outcome. So, for example, for the test instance X, you see, if we feed this into a black box model, we get a prediction of 1. Let's say it's a simple binary classifier. And then if you apply a feature attribution model, we could see how each individual feature value contributed to the final outcome. On the other hand, we have rule-based methods, which aim to define simple if-then rules that consistently lead to a specific prediction outcome. It's easy to visualize this as a sort of hypothetical decision tree, where you could go down and see which part of the tree applies to your test instance and see which prediction outcomes is a result of specific feature values. And finally, we have example-based methods. These try to suggest hypothetical virtual test instances, which might be very closely related to your original one, which have a specific effect on your prediction outcome. So, for example, we could change the second feature from a 1 to a 0, and then we see that our black box model inverted predictions from a 1 to a 0. These are usually quite intuitive explanations because they're easy to interpret due to contrastive reasoning. Next, I'll show you some of the most known XAI methods and how we've adapted them. So the concept Andrea already introduced, Shapley values were originally introduced to divide the total payoff amongst a group of players to reflect their contribution to the game. So the analogy he used was with the football players. So players work together in smaller groups called coalitions, and you compute their marginal contribution across all possible coalitions in the team. So we usually define it formally in the paper as divided with the formula with where you go over all different coalitions. Basically, this term here represents the marginal contribution of the coalition you're considering with the player and without the player. So the nice thing about SHAP is that it has basically a nice mathematical background, and it has a set of axioms that it has proposed, which all Shapley values should adhere to. So this is basically the mathematical theoretical foundation validating the existence of the Shapley value. So a few of these axioms are, for example, efficiency, which says that the sum of all of these individual contributions must equal the total payoff. You have the symmetry that if 2 players have the same contribution across coalitions, they receive the same Shapley value. And the dummy axiom states that if a player does not contribute at all, he's very lazy, he receives a Shapley value of 0. And there's a 4th axiom as well called additivity. So SHAP basically adapts this principle, this theoretical foundation, to explanations for molecular black box predictions. So as Andrea already said, the analogy slightly changes. Here, players are the input features. The game is the model prediction of the test instance, and payoff is the difference between the prediction and the expected value or the average prediction of the model. So SHAP calculates the mean marginal contribution of all feature coalitions for each respective input feature. However, as you can imagine, if you want to do this exhaustively, this is quite hard because it exponentially scales with the amount of features or your feature input space. So it's an NP-hard problem, which basically is computationally infeasible. So this is why for SHAP, especially for molecular explanations, we use approximations, usually for extensive sampling procedures. For example, this is an example for a Monte Carlo approximation, where you calculate the contribution of a feature across a few sampled coalitions or sample test cases. So one aspect to note is that, including for the SHAP, the Python implementation for the SHAP package, but also besides that, there's a lot of different SHAP methods that have been proposed and are available, including the method Andrea presented. you have model-specific methods that's basically SHAP implementations that are specific to specific algorithms and use parts of these algorithms to speed up the calculation or even allow for the exhaustive exact calculation of Shapley values. On the other hand, you have model-agnostic calculations, explainers such as the original kernel SHAP that are applicable to any kind of model that's largely being replaced by permutation SHAP. So how do we use SHAP generally for these molecular applications? So if you have an exemplary test compound, which you see here, we can create the binary bit vector, basically where a bit vector of 1 denotes the presence of a specific molecular substructure, and 0 denotes the absence of a specific substructure. So with this representation, we can then feed this into a black box model and get a prediction and apply SHAP. And then we will get a feature vector in the same dimensionality as the original vector with a set of feature values. Now, what's interesting is that, of course, you can get feature values or contributions of features for not only the fragments that are present, but also for the fragments or the chemical fragments that are absent in the specific molecule you represent. You only get a contribution or a Shapley value of 0 if basically the feature is completely redundant and was always present or absent in your training set. But of course, if you just look at this vector by itself, especially in the dimensionality of a usual fingerprint being a bit over 2000, it's hardly an explanation that cannot really be interpreted. So what we usually do is we map the feature contributions of the present features back onto the original compound, so we can then structurally interpret with our chemical knowledge how these specific chemical groups contribute. However, there's a bit of a problem to some extent with this kind of visualization, and that it only reviews the contribution of present features. And as I said, these absent chemical features can also contribute. And you really need to know the expected value of a model or the average prediction for this to make any sense. Another hallmark of SHAP, which is nice, is that you could aggregate over multiple explanations to get a sense of global feature importance. So, for example, for a test set with compounds, we could get the feature contribution of Shapley value vectors for each of them, and then we could aggregate over this whole matrix of feature contributions, and then end up with an array of basically global feature importance to model predictions. And then from this, we could sort the resulting array, and from the most contributing features, we could then locate what the encoded molecular substructure is, which is basically a nice way of showing which substructures contributed most globally to the model explanation. While this, again, is a nice explanation, there's a few problems with it, in that the aggregation part is not always so simplistic to do, because, as I said, for each individual prediction, the Shapley values add up to the difference between the expected value and the actual model outcome. Well, the expected value is always the same with an individual model, but the actual output probability, for example, is different. So each of these vectors add up to something different. So you need to normalize to some extent or take this into account. Additionally, you might want to calculate what are the most important features differently for absent and present features, which is also something you need to take into account. And while these molecular substructures, which would then be the most important bit positions, is a nice explanation, these usually encode very small structures, which again, from a chemical perspective, is not the most satisfying way to provide explanations. So therefore, I want to introduce a different concept for the Anchors methodology. It's a simple rule-based XI method. We try to define a if-then rule of specific feature values that always lead or guarantee a specific model outcome. So, for example, on the bottom right for you guys, left actually, for example, a test instance, we could define a rule where feature 1 always has to have a value of 0, and feature 2 has to have a value of 1, and then we always get a model outcome of 1 in about 95% of the cases. So that would be such a rule. And formally, we can define this as an anchor rule applied to test instance X is a rule for the outcome of 1, and for a distribution of perturbed instances Z, where the anchor still holds, so it has these specific feature values. The original prediction is held for a specific precision threshold percentage of times, which is usually quite high. So you want about 95% of the perturbed instances to maintain the original prediction. However, and this is quite a simplistic method and it's quite intuitive to calculate. However, for larger feature spaces, especially our representations, this becomes completely infeasible to calculate exhaustively because of the huge space of possible search pools. So we've adapted this to molecular anchors, or we term MoAnchor, where we try to identify specific molecular substructures that are responsible for a given prediction. So, for example, if this was a predicted molecule, we could identify this benzodiazine substructure, or what I've highlighted in red, to be solely responsible for this given prediction. So this approach has a few advantages by basically constraining the possible search space of rules to a few relevant chemical substructure combinations. We can calculate this positioning threshold exhaustively, and we can immediately return explanations in the form of chemical substructures, which increases the interpretability. So how do we do this? So for a given test instance, we would decompose it into molecular fragments using a specific fragmentation formalism, which always guarantees retrosynthetically accessible substructures. Then we can map these individual fragments back to the original feature presentation, which is, of course, easy to do with our fingerprints, but especially with molecular graphs. And then using this representation, we could identify all possible combinations of molecular fragments. So you basically, for example, in a simplified table with only 4 molecular fragments, we could generate a table like this, where a 1 denotes the presence of a fragmenting combination and the 0 denotes the absence. So then using our molecular representations, we can repredict these perturbed instances using the original model and try to identify a fragment that satisfies our precision threshold of maintaining the original prediction. In this case, this would, for example, be this fragment, with when it's present in a combination, always use the corresponding prediction. So here's an example of applying MoAnchor to a simple binary classification. And we also applied the original anchors methodology, which just works on the binary bit vector. As you can see, the original anchor methodology identifies 2 bits that have to be there to basically guarantee the prediction outcome, being 2 disjointed bit environments. And the original the new MoAnchor methodology identifies this larger chemical substructure to be solely responsible for the prediction outcome. So, of course, this is slightly easier to interpret. It also makes more sense from a chemical point of view. However, using one method or the other is basically a trade-off between inherent interpretability and the granularity or the precision of your explanation. However, since our primary task is to explain our predictions to chemists to make sure that they trust it, going for something more intuitive might be advantageous. One thing to note as well is that MoAnchor took a few milliseconds, I think, to run for this prediction, while the original anchors method ran a few minutes due to the huge size of the search space, so optimization. So we wanted to test this new MoAnchor method in a proof of concept study, basically to explain the correct prediction of isoform selectivity. So we created 3 data sets. Very simply said, for each data set, you have compounds that are active against 2 protein targets. However, some of these compounds might be more active against one or the other, based on a specific cutoff we use. So then we use the model to predict in a multi-class sense whether a given compound was not more active against one protein target or the other, or perhaps it was one more active against protein target A or more active against protein target B. And then we applied MoAnchor to these correct predictions of selectivity to see if we could identify any molecular substructures that will allow the model to be accurate in these predictions. So we tried to generalize these rules over entire test sets and thereby identifying these recurrent molecular substructures that yield corresponding predictions. So for this, we devised 2 specific metrics, being substructure occurrence, being the proportion of these tested compounds of a given class that contain the substructure, and the anchor occurrence being how often these substructures being present in these molecules actually also are identified as an anchor, so are solely responsible for this correct prediction. So as such, the anchor occurrence can never be higher than the substructure occurrence, And if they're equal, it means that if this substructure is present in a molecule, it is always solely responsible for the correct prediction of this molecule. So as you can see for these different isoforms in different prediction tests, for example, the P10 delta, so marked in blue to your left, you see that about 30% of the test set contained this molecular substructure, and in almost all of them, it was solely responsible for the prediction and was identified as an anchor. And for some of the other selectivity predictions in isoforms, you see that it's basically whenever this specific fragment identified small anchor was present, it was solely responsible for the correct prediction, showing the utility. Next, so we wanted to determine whether there was actually a relationship between these explanations and actual isoform selectivity. As Andrea said, we have to be very careful with the term causality here, because machine learning is inherently based on statistical correlations between the input features. So you should perhaps refer to causal ML for actual causality exploration. But we looked up these molecular fragments. We found that often led to correct predictions. And in literature, we found that these specific structures were actually generally used to modulate isoform selectivity because they bound to a specific region of the protein. So in this case, there was actually a causal relationship between the explanation and the actual biological process of isoform selectivity. Of course, you can still not really say the model learned chemistry, as it were, because, of course, these fragments are used to modulate selectivity, so they're overrepresented in the data, which the machine learning model learned, and which is reflected by the explanation. But it was nice to see either way. So a second type of methodology I want to show you is counterfactuals, which is a different example-based method to describe a causal situation in the form of had X not occurred, Y would not have occurred. So in terms of molecular predictions or ML predictions in general, this is often defined as the smallest change we can make to a given test instance so that the prediction outcome changes. So for example, we could change the second feature from a 1 to a 0, and we can see that the prediction outcome changes. So the quality of such counterfactual explanations are usually measured in terms of achieving a desired outcome with as little feature changes as possible. So for molecular design, we can adapt this definition to molecular counterfactuals, where we aim to achieve a different or pre-selected decision model outcome change based on the smallest chemical modification we can make to a molecule. So this, as you can see from the explanation I showed here, this is usually quite nice because it's very visually representative and intuitive explanation. And it's also a very easily verifiable hypothesis in which you could, for example, test a new molecule to see if this kind of counterfactual approach is actually correct. However, usually the most machine learning-based counterfactual methods achieve counterfactuals for random feature perturbation, which in our case doesn't really make sense because you would get completely nonsensical molecules. So we need a way to model the local chemical space around a given test instance, so we can draw hypothetical instances, repredict them, and identify counterfactuals. So for this, we actually adapted a very simple chemical principle, which Andrea also introduced, being that of the core and the substituent, that a lot of the chemical data, a lot of molecules share a similar core structure, but differ just by one simple substituent, which I've circled in red here. So what we did is basically isolated all core structures from a given data set, and then we can recombine these core structures with a list of substituents. In our case, we used a list of substituents which are highly recurring in chemistry to create new hypothetical test instances, which we can then use the black box model to repredict to see if there's any instances that change prediction, and then we can identify them as counterfactuals. And then if you have a large population of these counterfactuals, we can then basically rank them on how big the change was, either through molecular similarity or change in molecular weight. So we also applied this to a proof of concept study, which was a multi-class classification scheme in which models had to predict whether a given compound was active against one of 6 different proteins or kinases, which are a specific type of protein. And here you can see that basically for two correct predictions, with one predicted correctly as class 1 or class 2, how these different substituents lead to different class introversions, and even for a specific prediction, how two different substituents can lead to different class introversions, which allows you to study the decision borders between classes. But you can also look if there's a specific core structure that really often lead to the generation of counterfactual. And these are core structures that are probably right on the edge between these different prediction classes. And then you can look at the edge cases and define, for example, the smallest change, so the classical formulation of which we can make to the core to make a class introversion, or we can identify a set of substituents that make the model the most confident in the newly predicted class, which are usually, of course, larger changes we can make to the molecule. Secondly, we can also look if there's any substituents that very often lead to the generation of a specific class counterfactual. And then we can conclude that these recurrent substituents are likely very important for the specific type of specific class prediction. As you can see, this often constitute larger changes or larger substituents, which would have more effect on the outcome. But it's also nice to see, for example, with class 2, that a lot of these substituents that are recurrent are substructures of each other, meaning this kind of structure is actually important to the model decision. So a few concluding remarks and future perspective. So I'll show you how we can adapt existing XAI methods to provide more chemically intuitive explanations, thereby going from simply showing data relationships of the patterns and move towards interpretability, where these explanations are more understandable to us as humans, especially the end audience being chemists. So some of these resulting explanations, especially the counterfactuals, could hypothetically be verifiable hypotheses, which you get used to guide follow-up experimental studies. And giving the right scientific rationale and right study setup, you can explore causality given the right model explanation method. So a small future perspective here, and I think Andrea already alluded to a lot of it, but XAI is naturally a supportive field to the larger field of machine learning. So naturally, we're usually a step or two behind the very quickly adapting methods of machine learning. So one thing we still have to do for molecular design, which has only been experimented with a bit, is properly using attention quantification for transformer models, also when they're applied to a simpler task like regressional classification. However, the nice thing about attention is that it's inherently used, so it doesn't require that much follow-up calculations. But there have been some worries whether you could really equate attention with actual explanation, but that's a whole article line that you might read at your behest. Otherwise, you could also look at different methodologies like gradient-based explainers, like integrated gradients, which rely on a backwards pass to calculate explanations. And one of the big things we still have to work on is explainability for these generative chemical language models. So as I said, for sequence-to-sequence tasks, since sequences, especially in token-by-token transformer models predictions, this is still just a probability distribution. So most feature attribution methods could realistically still be applied, albeit, for example, SHAP with a lot of approximations and adaptations to the methodology. Could also do this with attention. And I think one of the most promising ways of exploring the learning characteristic of the more complex models is simply augmenting your input representations. So with generative chemical language models, in this case, this is an example of what it would look like if you would quantify the attention. So if this was a simple task of having one input sequence represent the molecule, and you want to have a specific characteristic modified, and you have a different output sequence, you would visualize the attention of the constant tension between these two molecules. It would kind of look like this. of course, as you can see, this is not necessarily very interpretable. So we would have to either aggregate this in a better way of create different visualizations for this to become interpretable to a chemical audience. And as I said, another way to study the learning characteristics of models is by selectively augmenting parts of the data. And so, for example, here, this was done also within a group where they studied the learning characteristics of a protein language model, which, based on the protein sequence in terms of amino acids, was used to suggest new compounds. And usually the way you evaluate such models, because not all of us have ways to have follow-up experimentation to validate the molecules, is basically to see if these models refabricate any known active molecules, which is what it wasn't trained on. And so you see that the model was more or less able to do this for this specific protein, but then they wanted to know, why is a model able to do this? So what they did was masked off specific parts of the input sequence from which, for us as chemists or biologists, it wouldn't make sense for the model to be learning from. So, for example, the binding site is a part of the protein where the compound actually interacts with. So you would think this is important for the model to learn from, to know which compound it has to suggest to be active against a specific protein sequence. However, what they found is that they masked off these biologically plausible regions that didn't actually have an effect on the performance of the model. So this kind of suggested that these models are learning from global sequence similarity and actually sort of regurgitating compounds that they've seen before, but they're not actually learning any biological context in the way we would hope them to do so. So it's just a different way of evaluating your models that does not necessarily rely on XAI methods, but still is a very good way to go forward, I think. So thank you.
Great. We have time for questions here in Bonn and in Dortmund and online.
For you, it's not completely solved how to map attention maps to the input?
Yeah, so theoretically, there's different ways you could go about it. For example, you could do it in terms of what you would do with images with superpixels, where you could do it, for example, molecular fragments instead of doing it on token by token. So you would get a more intuitive mapping, but this is also not, or it hasn't been experimented with yet. Alternatively, what you could also do, basically average the attention maps on a atomic basis and then map it back to the original compound. But this is also, it's whether that then constitutes a good token by token explanation, I haven't seen yet. So it's still a bit of an open problem how to adapt this. So if you have any ideas, of course, we welcome that.
No, I mean, it's like much harder than for images, right?
Yeah.
Okay.
So I have a question. This is a very non-technical question about the counterfactual approaches.
Mm-hmm.
I mean, I've come across these in other areas of XAI as well, right? And there's always something a bit odd to me about them, right? Because I mean, suppose I'm late to a meeting, and somebody asks, "Well, why were you late to a meeting?" And I say, "Well, if the train had been on time, then I would have been here on time," right?
Yeah.
This is a sort of a strangely indirect way to answer the question of why I'm late, right? I'm sort of hinting that I'm late because the train was delayed. Right? I'm not quite saying it, right? I'm doing this really indirect thing. And so there's that kind of aspect of these explanations, right? It's like you want to say what made the model give the outcome, but you don't quite say that. You say, well, you know. And the other thing that's strange is that given the way the models work, you have to say something, not just, well, if the train had been late, you have to suggest some other thing. So, that if I had driven a car instead.
Yeah, exactly.
And that might not be right? It might be like, even if I had driven a car, it would still be late, right? So the difference between saying, if I hadn't taken the train, I would be late, but it's not the same thing as saying, if I had driven a car, I would have been on time, right?
Yes.
So how do you deal with those things in this area, or just live with them? I don't know, but
that's always a good approach. But well, it really depends on what you want from your explanation, right? So I think counterfactuals, a large base of why they're used regularly is that the fact that it's based on contrastive reasoning, which from the area of psychology and human studies is said to be the most intuitive way of explaining new new explanations among humans, because we tend to also do it when we contrast something different when explaining a specific observation. So I think as a method, it's less based on trying to diagnose your model, which would make more sense to do with feature attribution-based examples. But it's a way to maybe also convey trust in your model by showing where the decision boundaries are. And whether you trust your model for this to be correct, that's a whole different perspective.
I guess that makes sense. I mean, that kind of, to me, suggests that there might be more value in like sort of families of counterfactuals for each case, right? So to make these decision boundaries, say, well, if we had done this or this or this or this, then the outcome would have been different. And so that gives you a fuller sense of why this.
This is also a whole part in the evaluation of counterfactual approaches, is that one of the things it said is to be important is that you not only generate one counterfactual, but generate families where you can have a complete sort of, in terms of chemical space, you can identify the edges of the chemical space where the explanation applies to.
Yeah, I see, that makes sense. Okay.